Today I'm going to talk about Foxp3 instability. We think probably this instability can help Treg to distinguish the self and non-self antigen. The most fundamental question for immunology is how to distinguish the self and the no-self. Every student has learned our system can do the perfect thing. We attack the pathogen but leave our cells alone. But how this achieved? Just basic on the Bennett clone selection model. Every animal has a cell called the lymphocyte. The lymphocyte is reprogrammed to recognize a particular antigen. Once they encounter this antigen, the lymphocyte will stimulate, proliferate, and differentiate into an effector cell and produce a lot of effector molecule like antibody, then will kill the pathogen and stay our life. They also mentioned early exposure to the antigen just at birth, early tolerance. The cell was depleted in thymus or the bone marrow, then the cell just died. That's pretty black-white model. So if you have the self-antigen, it's just gone during the thymus selection. But this survived cell actually is just based on this model, it should be not self-reactivity. But this white-black model can raise a lot of questions because we are not staying forever. We are changing. We are growth. When the organ getting older, they produce a lot of new protein, but this new protein never get react by the immune system. We eat a lot of food, of meat, but they are not going to react either. The mom didn't reject the baby because there should be MHC in match, but we never get a response. But we still cannot make a reaction, just inject the protein in our body. We need another thing we call the adjuvant. So what's the adjuvant is? We cannot reject the tumor. For sure, they're mutant, they're not self-protein. We cannot, even we have a lot of autoreactive T cell in our body, but normally the T cells normally don't cause a lot of trouble. So this is Polymedicine Singer writings in a review in Science. There is a lot of questions for this original self-non-self model. Yeah, then the model have to modify, to switch, to adapt to understanding in the 1969, the first adaptation actually is the B cells need the T cell help. If they just receive signal 1, they just die out until there's some T cell helps the B cell, they get survive and produce antibody. Then in 1975, the second modification, the T cell also need the co-stimulation. Just signal 1 didn't provide the enough signal to drive the T cell to activate. You need a second signal, normally we call the B7 or CD28 in the T cell. Then the cell get fully activated, then they proliferate and prefer their function. Yeah, come to the 1989, Chairman, we have proposed a new model. They said this APC is critical. If you don't have this APC, it's just a DC, probably the most important antigen presenting cells, not enough to produce a B7 signal. It cannot co-stimulate the T cell. So only you have this toll-like receptor signal, then trigger the B7 after activation, they go drive T cell to activate. Further, Polymedicine Singer have proposed a danger model, although a lot of people don't like her. But I think it's quite makes sense where a lot of H-Hawk protein actually can produce a similar signal like the toll-like receptor. No matter what, this is a basic is a two signal model. We have a signal 1 is from non-self, the signal 2 is from pathogen or alarming, whatever you need the CD28 to make the T cell to act. If you don't have CD8, they'll stay quiet under tolerance. But the life always more complex than we thought. When Jeff Bluestone have crossed the B7 knockout to the NOD, it's the type 1D autoimmune model. Most people will predict that then the mice will survive and protect from autoimmunity. But the case is a complete opposite. When you get rid of the B728 in the NOD background, the mice get much sicker. They die like 12 weeks or 15 weeks, just get a huge autoimmunity. It's not make sense at all for most of immunologists until you figure out in this setting, when you knock out the B7 CD28, another important cell is gone, that's Treg. Treg is not a new concept. In early 90s, Richard Gasson had proposed not every T cell have their effect function, but some T cell have a regulated function they call suppressor T cell. Has been intensive study over a decade, but in the mid-1980s, the field totally dead because no people believe suppressor T anymore. The major reason is don't have a reliable marker, can distinguish the suppressor from normal convenient cells. The modern will come from the Shimon-Sakaguchi study in 1995. Shimon-Sakaguchi was trained with Nishizaka as the professor who developed a very interesting model, day 3 synectomy. When you react the thymus from day 3, the mouse just develop all sorts of autoimmunity. We don't know they don't know what's going on in the stage. Shimon Sakaguchi enters Nishizaka's lab in Aichi Cancer Center. It's not a very famous institute, but he's retriggering by this really interesting finding. He believes there are some regular population can develop from the late stage of thymus. It takes almost 20 years. Shimon Sakaguchi found a marker, it's a CD25, actually can distinguish the normal cells and the regular population. He used a CD25 molecular antibody with a complement to get rid of CD25 positive cells and inject to the new mice. The mice got all sorts of autoimmunity. That's called systemic autoimmunity. So he proposed a new cell type. It's called a regular T cell. We'll call it Treg right now. It's not a suppressor T. The suppressor T normally we think is CD8, but this is CD4 positive cells. Treg is just a small population, really tiny. It's about 10% of in our periphery CD4 population. They also found in the thymus, only about 5% in the mature single positive thymocyte. They also exist in mice, rat, and human. It's weird phenotype because they're energetic in vitro. It's hard to react to any antigen stimulation. They have very strong in vitro and in vivo suppressive activity. The basic phenotype is just like CD25 high, 45RB low, CTLA4 high, and the GITR high. This phenotype is just like the active cell, to be honest. So that's why even Shimon published the paper in, a lot of people still don't believe this, because it's just an active cell express a similar phenotype like this, the Treg he mentioned. Since changing to 2001, Bruno and Fred Ramsdell have cloned a new gene called Foxp3 from a weird mice we call scurfy mice. This is an X chromosome induced autoimmune mice. This mice just get weird autoimmunity in 3 weeks after born. Mice just die really quick. Fred and Mary had to take a long time to figure out what's going on in these mice. They found a gene called Foxp3 actually mutated in these mice. They made a transgenic mice and found actually they can rescue this scurfy mice. The mutation also found in human, this weird disease we call Apex syndrome. This, most people cannot survive probably get lethal autoimmunity just before 1 year old. But right now we can do the mixed bone marrow chimera or something like that to make their patient to alive. Foxp3 is a transcription repressor. If you overexpression Foxp3 in T cells, it just suppress the T cell response. They have this fork head domain in the C terminal and the leucine zip and the zinc finger domain in the middle. In 2003, three group actually linked the Foxp3 to Treg. Sasha Ludensky first found the Foxp3 is expressed in CD25 population. It's not in the active T cell. Most marker actually found in the active T cell, but the Foxp3, at least in mice, is not expression in active T cell. This is true in real-time PCR and the Western blot. Sasha's group also made the GIP knock-in mice to show the Foxp3 expression in single cell level. They actually overlay with the CD25 population. They also noticed the Foxp3 positive cell indeed come late. If you just check the day 3 thymus, the heart had any cells in this stage. They are divided much later than the conventional CD4 T cells. Sasha also made the knockout mice, just get rid of Foxp3 directing mice. They have the similar phenotype, just like Skirfin, had a really huge lymph node and spleen, had all this infiltration. That's because the T cell get activation, they just attacks their body everywhere. The mice just die 3 weeks old. Of course, if you transfer CD25 positive cells in these mice, the mice can survive much longer. Sasha Ludensky also have another, I think, really nice experiment to demonstrate cells critical express the Foxp3 DTR. They use a knock-in strategy, put the diffusor toxin receptors into the mouse cells. Mouse normally not react to this toxin because they don't have a receptor. Once they express, use a Foxp3 drive this DTR expression, the mice just like no more like similar. But if you treat with a diffusor toxin, the cell just dead. Then the mice have a weird phenotype, which is develop autoimmunity in 2 weeks. Mice just died. In the other hand, Shimon Sakaguchi have overexpression Foxp3 in T conventional cells. They show this even in the normal T cell, if you overexpression Foxp3, they gain this kind of suppressor activity in their colitis model. So summary is all this evidence definitely demonstrates Foxp3 is a critical regulator gene for Treg because their expression is really strict in the Treg population. Their knockout have this apex-like syndrome, which can be rescued by CD25 positive Treg. If you just get rid of Foxp3 cells, use a DTR strategy, the mice just die like 2 weeks. We also can use overexpression Foxp3 to make the non-Treg to become the suppressor in vitro and in vivo. So all this evidence bring this Foxp3 is a really critical regulator and Treg is really critical. When I joined Jeff Bluestone lab, I just wanted to do more Treg work. I just hate to do all this in vitro suppression assay. I don't think that make any sense. So we start a mouse model with just generate the Foxp3 cream mice. When we cross with dicer, flux dicer, we made the first condition knockout, the mice just die like Skirfin mice. This is also the first conditional setting can phenocopy the Skirfin mice. Yeah, I already know that already. Mary Berklow and Fred Ramsdell and Simon Sakaguchi win this Nobel Prize this year. But the big question still remaining in this field. All this really basic question, I don't think this really answer if the suppression need antigen specifics, is the suppression need cytokine, is the suppression need antigen presenting cells. Actually, we not know on the answer, but we have some clue, maybe some evidence. For this antigen specificity, we kind of know that already. People have published this in vitro data. If you put the Treg and the T conventional cell with the same TCR, put the peptide together, they have strong suppression. You can put the different TCR with a different peptide, but unless they're active, they still had a good suppression. But if you only add the peptide to stimulate the conventional cells, will not have any suppression. So we know at least the Treg need to be activated. Once they're activated, they don't need this specificity anymore, but at least they need activation. In vivo evidence come from also from Sasha Roden's labs. They made the Foxp3 pre-mice flux TCR alpha. The mice, the Treg cells just stay in naive condition. They're not die, but they're not active. They're just stay resting condition. The mice actually develop a similar scurfing autoimmunity like the Foxp3 knockout. So both on in vitro and in vivo, the Treg need antigen to be activated. But once they activate, they may not need the antigen anymore to do the bystander suppression. So this is a basic summary. When the T cell get a naive Treg cells, when they see this antigen peptide, most we think this is self, they will probably get activation. They may be downregulated CD25 in their surface. They gain the CD44, they change their surface marker. They produce like 300 different genes, including IL-10, IL-35, CTLA-4, IL-4, to commit the suppression activity. Okay, we also have some similar finding when we knock out the NOC4L, this ribosome regulator. We see this, once we knock out this gene, the mouse has like scurfing phenotypes that must die really quick. They also cannot active in this stage. Similar finding, we knock out the MEL1, it's another epigenetic regulator in Treg. We also get a similar phenotype that's much less severe. Interesting is Foxp3 actually regulate T cell signal as well. When we see this Foxp3 mutation, a splicing form in human, we generate the mouse to mimic this human splicing form. Actually, we found the Foxp3 combined the BATF promoter very strong, the suppress their T cell response. So Treg need the TCR, and the Foxp3 also suppress Treg. They kind of had that kind of ritual relationship. How Treg works, there are more than 15 or 20 different mechanisms. This is a small map to summarize some part of this. Treg can produce suppressed cytokines such as TGA-beta, IL-10, IL-35. They can produce granzyme and perforin to direct kill the cells. They express a very high level of CD25. This makes a perfect sponging to shut all the IL-2. There's a really strong competition for the CD8 cells. They also produce CD39 to degrade the ATP. Of course, Treg can connect with DC very well. They can paralyze DC or use the CTLA-4 or some other mechanism to suppress DC. So all these different mechanisms exist. We just wonder, is the Treg really like Leza? we call the bird have 3 head, 3 arm, 6 arm. The altered Treg have this all this function together, or these just have 2 different subsets. We made the IL-35 reporter mice actually found this interesting. The IL-10 producing Treg and the IL-35 Tregs is separate. They're not the same lineage. When we treat anti-CD3, we can enhance IL-10 production, but not the IL-35. When we use a blimp knockout, we can complete blocking the IL-10 production, and we not infect this IL-35 production. At least we think we have 2 different or couple different Treg exist. We also figure out that the T-cell signal strength is critical for this IL-35 Treg and IL-10 Treg production. Strong T-cell signal can drive the BLIMP1 activation, then drives all this effector molecule and the migration molecule to like the Treg to go outside, where the IL-35 Treg potentially expresses CR7 as the Treg stay in the lymphoid organ. So there definitely have different subsets for Treg to confirm their suppressive activity. Another interesting topic is Treg specificity. We probably can divide the Treg into big 2 different subsets. One we call the thymic drive Treg because they're from thymus. Another we call the peripheral drive Treg. They're largely sitting in the gut when the strong TGF-beta signal and IL-2, they can change from the T conventional cell induce Foxp3 expression, and they called the peripheral Treg. Most of peripheral Treg probably recognize the foreign antigen. That's well known. But for the thymic antigen, they come from a thymus. They need a strong T-cell signal in the thymus to get the Treg develop. But they also come out from the thymus to the peripheral lymph node and the organ. They can tag all different antigen, including self and non-self. They can. But whether they have this kind of specificity issue. So another key question is, can this thymic drive Treg to distinguish self and non-self? Because they have this highly diverse TCR receptor repertoire, just like our T conventional cell. Can they tell the self and the non-self whether they can respond differently from this self and the non-self antigen? We get some clue from our fit mapping study. When we generate this Foxp3 cream mice, we breeding with the Rosa YIP, the reporter mice. We did a fit mapping study in this setting. We found some Foxp3 cells actually can lose Foxp3 expression and downregulate this Foxp3 and change to effector. We called this cells as we call the X Foxp3 or X Treg cells. It's a consequence of Foxp3 instability. We don't know for sure why they shut down Foxp3 in the Treg, but they are not Treg anymore. They don't have a suppression activity. They actually has a phenotype like active memory T cell phenotype. They produce a lot of inflammatory cytokines such as interferon gamma and IL-17. When we transfer the autoreactive X Treg cell, it can cause really rapid type 1 autoimmunity, like T1D. They just can kill the islet that's strong like the effector cell. There are a lot of studies have showed this X Tregs come from all the different autoimmunity model. The paper have been published and cited quite a lot, but still a lot of people don't believe this, including Shimon Sakaguchi, including Sasha Lubensky. Sasha said our cell system is not perfect. They use rare ER2 Cree mice. They don't find this. Shimon Sakaguchi said, because you are using Foxp3 Cree, so some uncommitted cells maybe change to X Treg. So we decided to make another mice, just use a back to the transgenic strategy. We got rid of this CNS1, it's critical for smart binding site. When we get rid of this smart binding site, the PTReg induction is completely gone. So we can map the only thymic drive Treg. Anyway, we start a new mice. We did the confirmation because they're thymic drive Tregs, neutropenia high, Helios high. When we get on this population, we can find our reporter is only expressed in the thymic drive Treg, but not the peripheral Treg. We also did an in vitro experiment. We treated with TGF-beta and not induce the Cy1 and the Cre expression. There are also mapping the mature Treg. When we sorted this YP cells out, we did the PSTR stability confirmation. They're totally stable population. It's true. Just like Simon Sakaguchi and the other person said, thymic drive Treg is much stable. We cannot detect a huge population loss Foxp3 like our adrenaline mice. They're much lesser. But when we transfer these cells into empty the host, they don't have IL-2 in this mice, they start to lose Foxp3 and change their phenotype to produce interferon gamma, and they also can come from change to TFH cell to have B cell to produce antibody. We also found the IL-2 because there is a really critical survival factor for Treg. When we neutralize with neutralizing antibody, the Foxp3 lineage is much getting unstable. They can produce a lot of X-Treg cells, but it's not ICOS doing this completely different story. When we broke in ICOS ligand, the Treg cells actually getting more stable. Because ICOS, the downstream signal is the PI3 kinase. When we overexpressing PI3 kinase signal in this Treg cell, they lose Foxp3 as well. Similarly, if we get on this TFR, the cells go into the gem center, they get really active. We found a lot of cells actually lose Foxp3 in this setting. Cut the long story short, we found, we remade the mice. This mice only mapping the thymic drive Treg. They're really stable in the normal condition, but when we found once they go into the gym center, they're probably lose the CD25 expression, and they enhance the ICOS signal, then lose Foxp3 expression. So basically, from this conclusion, we made another hypothesis, just for antigen actually can drive this Treg to unstable. That's we cross this our reporter mice with OT2 with T-cell transgenic mice specific for OVA. When we immunize the mice with MP OVA, the antigen, the Foxp3 just gone a lot. The downregulating Foxp3 induces strong loss of Foxp3 expression. A good part of this system, we can detect different affinity impact on this instability when they use all sort of this different peptide to induce a strong and weak stimulation in Treg. We found once we get a really strong activation signal, they lose Foxp3, but this weak signal can keep the Treg stable. All this study come from the weird setting because the OT2 Treg is not exist in vivo, whether we can get cells from in vivo Treg. So we immunize the mice with MP OVA, sort the TFR population out, then do the single cell sequence. Luckily, we have one clone actually can react to OVA. This have a really strong affinity to OVA and a really strong reactivity than the OT2 mice. Interesting, when we generate this, we call OTReg1 transgenic mice in we found the mice actually have the negative selection. That means the T cell do have the self-activity to themselves. When we take this setting, we immunize mice again with over, we found a similar finding like the OT2, the Treg. When we gave them the TFR population, the Foxp3 just disappear a lot. We did also by using the tetramer study to study the OT2 polyclone population. This is a vaccine stuff. When we immunize the mice with NP, this peptide from influenza virus, we use a tetramer to get the antigen-specific T cell. We found the antigen-specific T cell can lose a lot of Foxp3 production, but not in this tetramer negative population. So here's the summary. We think that in the thymus, when the T cell signal to the self-ligand can get a huge range, the strong T cell activation can cause their death, cause negative selection. But the Treg, the middle range to the self-activity can survive and change their phenotype to Treg, the upregulate the Foxp3. Because once they go to the periphery, because their self-activity was removed by negative selection, we probably don't have this really strong self-activity in vivo, in periphery. They only encounter the middle range of self-activity. They're probably good enough for their activation and confer their suppression activity, but not make the Treg unstable. But non-self antigens, such like OVA and NP antigen, they can cause a very strong T cell signal. They may combine with all this information like toll-like receptor engaged, like IL-6, IL-4, all this stuff together maybe makes the Treg unstable. They're not have any suppressive activity. They actually help the immune system to account this pathogen infection. I would like to thank all the lab members, particularly Zhongmei and Liping did all this XTREG stuff. I really thanks to the grant and all this collaboration. Thank you so much. I'll take this question.
谢谢周老师非常精彩的报道。我想，就是后面那一部分就是您提到不同的抗原，它可以诱导这个 X T 的这个 Treg 项目变为 X Treg。它这个是一些抗原特异的去驱动它像做这个 TH1 类的或者 TFH 类的这样的途径吗？还是说它也需要其他的一些 DC 细胞的帮助去让它变成不同的？
我们实际上看过 TH1 和 TFH 实际上都会诱导这个 Foxp3 丢失，所以这个分化好像不是一个特别重要的因素。但是 TFH 我认为可能会特别容易丢失，因为在 James Center 里面它没有大量的 L2 反而是对于 Treg 的稳定是一个至关重要的因素。你一旦没有这个东西的话，那个 Treg 就变得不稳定，变得会死亡，丢掉 Foxp3。所以我认为可能是在 TFH 这步骤，就是调控 B 细胞里面，可能这个过程变得尤为重要。但是
B 细胞跟 T 细胞的
interaction 但也有可能有影响，对。但 TH1 实际上我们也看到类似的现象。我们如果 get CXCR3 positive 的那个我们叫 TH1 型的 Treg， 我们也看到很大量的丢失。所以可能不见得是一个就是这个分化特异性的事件，只是一个就是亲和力加上这些 inflammatory cytokine 的问题，可能是最主要的。
谢谢您的精彩报告。我比较感兴趣的是这个Foxp3 它不是那个不太能稳定嘛？
对。
然后这不稳定性的话，是否有针对它的一些靶点来研究一些不管是化学小分子还是一些抗体什么来调节它的稳定性，从而来调节它的
对对，这个非常好的问题啊，可能没有时间展开啊。这个现在实际上抗肿瘤的药物治疗好多是靶向 Treg 的稳定性，可以让它变得不稳定以后，实际上会促进这个肿瘤的消退。这个最著名的可能走的比较靠前的就是 EZH2的那个 inhibitor，那个在肿瘤领域上已经推的比较靠前了。还有一些其他的，反过来说，你在体内打上白加二，实际上不光是促进这个 Treg 的数量增加，它稳定性也会变好。所以这是可能做自身免疫性的疾病的一些应用，现在也在展开。包括这个 CTLA-4的抗体的一些治疗，实际上现在也有一些这个报道说，它实际上可以让这个 Treg 变得不稳定，最后来这个终止掉 Treg 的这个抑制功能。当然，这个 Foxp3 丢失，我们实际上认为是最终的一步。在这之前，实际上已经有很多时候，它这个 Treg 是失活了，就是包括好比说，现在有一些人讲的这个 Treg 在某些环境可以产生因特菲龙伽马，这个上我们考虑这些细胞可能已经是这个 Foxp3 丢失的前提了，因为它如果有很强的 Foxp3 功能的话，Treg 是不会产生这些就是这一类的细胞因子的。
谢谢。
这个我再问一个学啊，就是你这个结果现在就是提示，就是 Treg 的稳定性可能比 Treg 要更稳定一些，是吧？
对，是的，啊，
就是它的分子机制是什么？
分子机
然后
制，
另外一个就是在这个衰老啊或者一些慢性炎症的过程中，那这些 Treg，因为你现在是在 OVA 那个 specific 系统里面看到啊，在一些这种自然的条件下，衰老啊病理的一些条件下。
对，明昭这个问题太好了。对，这个就是实际上我们做过衰老的动物，就是衰老以后，这个 Treg 的稳定性会变差。Treg 就是这个耗子的，它的会产生更多的这些这个炎症反应。这个里面的原因我们现在还不知道，但可能有两方面，一个是像刘光辉他们做的这个会有一些病毒，像这个到了老年的时候它会释放出来，这些病毒会不会 trigger 这些相应的反应，这个是一种可能性。还有一个就是说老年化以后，它的这个 heterochromatin 的这个 control 可能变得不是那么紧密了，它可能会露出来一些东西，它就变得这个 Foxp3 的这个。表观是一个调控 Foxp3 稳定性的一个最重要的因子了，它在第二个 CNS2 上面有一个 CPG 的 motif，那个东西的话，好比说你在这个 Treg 里面敲掉这个 TAT2，它就会完全变得不稳定，甚至在这个 HAT 的这个老鼠里面，它甚至都会都有很明显的非正态。因为那里面虽然有50%正常的这个 Treg，但是你敲掉以后，它这个 Treg 细胞实际上变成 effector，它也会致病。所以我们觉得那个表观可能是最后的控制的一个最关键的元件，当然中间代谢和表观有可能有一些 link 在这里面，但是我们确实还没有做这方面。
好，谢谢。最后一个问题啊，
有个小问题啊，也是跟 Foxp3 相关的。其实，看到一些文章报道，其实我不清楚现在的一个比较统一的观点是，这个 Foxp3 表达是 CD4 比较 specific， 因为有一些文章里报道在人的这个临床样本上看到了 CD8 阳性的 Foxp3 阳性的细胞，而且我们单细胞数据里是有这样一群细胞的，但是我们好像在小鼠里面并没有看到类似的这个细胞亚群。我不知道这个 Foxp3 您现在认为它是 CD4 specific？
就说我们自己的研究来说，可能绝大多数是 CD4 阳性的，但确实是有一些这个 CD8 也会表达 Foxp3，就特别是我们用 Fate mapping 去做的话，它可能没有 Foxp3 的表达，但它确实是经历过这个 Foxp3 的这个经验的，那些细胞好像会更多一点点。在人的临床里面比较复杂，人的那个调控和小鼠还不太一样。最近可能那个Alex Masson 他们有一个这个文章，就专门在做这个事情。实际上就是做人和小鼠的那个表达调控到底有什么区别，有些 enhancer 什么东西好像还不完全一样。人的细胞在某些条件下，它可以有一个 transient 的这个 Foxp3 的表达，但是这个争议比较大。有的人在说是抗体的这个问题，还是这个检测不是那么特异的问题，这个确实是存在的。还有一些在一些特殊的脏器，好比说在 IEL 的这个环境底下，这个 Foxp3 也会丢失。 那些细胞，我认为产生 CDA 就不那么奇怪了。那个确实我们也看到了，确实是会这个 IEL 的细胞在丢掉 Foxp3变成一个这种怪怪的细胞，我们也不知道是什么。现在确实是有
谢谢。
Science supports healthier, happier, and harmonious life.